In today's video, we're going to take a look at inheritance for A-level biology. If you see a certain term on this page that you want to skip to, take a look in the video description below as I've listed all of the respective timestamps. Otherwise, without any further ado, let's get into this information-packed video. So the first term that we're going to start off with is locus. Locus is the position that a gene has on a chromosome. What is a gene? A gene is a sequence of DNA nucleotides that code for the synthesis of a particular polypeptide. Now that polypeptide can be an enzyme itself, or the polypeptide can form part of a larger protein, like an antibody, for example. An allele is a variation of a gene. So if we think about peas, for example, the gene for pod color, they can either be green or they can be yellow. So the allele would be that either the pod color will be green or the pod color will be yellow. So it's a variation of the gene in this scenario being pod color. Dominant alleles are alleles that mask the effect. Therefore, recessive alleles are alleles that have their effect masked by the dominant allele in the phenotype. Okay, so dominant and recessive, we're looking at the phenotypes. Codominance refers to when two alleles contribute to the phenotype of an organism. Linkage refers to when genes are closely positioned on the same chromosome. And when they are positioned very closely to one another on a chromosome, they are linked. And this linkage means that during crossing over, because those two genes that are linked are very close to one another, they will have a higher chance of being inherited throughout generations together. Okay, so there's a smaller chance of linked genes being separated during crossing over. A test cross is a cross that we do to identify the genotype of an unknown organism. Now, we will get our sample organism, and normally we will get a homozygous recessive individual. First filial refers to the first generation of organisms from two parents, and second filial refers to when we interbreed those F1 organisms with one another, and then we get that second filial, the F2 generation. Phenotype refers to the physical makeup of an organism, so the physical traits, P for physical. Genotype refers to the genetic makeup of an organism, G for genetics. Homozygous refers to when we have two alleles that are the same. We can have homozygous dominant when both alleles are dominant, and we can have homozygous recessive when both alleles are recessive. Heterozygous means that we have two alleles of different types. So it can be a dominant allele and a recessive allele. So as we discussed before with the pod color of peas, we can either have a green pod color or a yellow pod color. In other genes, like for example in the eye color of humans, there are numerous different variations of that gene. We can have blue eyes, we can have brown eyes, we can have green eyes, hazel eyes, all of these different variations. So all of these different variations of that eye color gene results in multiple alleles. So for the pod color of a pea, there were only two different alleles. It could either only be yellow or it could be green. In human eyes, we have multiple different possible phenotypes. We can have green eyes, blue eyes, brown eyes, etc. So that is multiple alleles. So in this test cross example, we're going to be taking a look at the gene for pod color in peas. Now, in peas, a green pod is the dominant allele, and the yellow pod color is the recessive allele. And we've described this, right? We've annotated this with a capital G for the dominant allele. So all the time, if you have the dominant allele, it's going to be the capital letter. And then the lowercase letter, lowercase g, will be the recessive allele, which in this case is the yellow pod. So when we go ahead and we do our test cross, the first thing that we have to write down is the parental, oops, let me just change the whiteboard marker. So the first thing that we have to do is we have to write the parental, so parental phenotypes. Now in this test cross, we're going to cross a green pea, so a green pea with a yellow pea. Okay, now remember, phenotype refers to the physical makeup. So these peas are going to be, one will be green, that's its phenotype, and the other one is yellow. The next step in the test cross that we have to do is we have to write the parental genotypes. So parental genotypes. Now, genotypes, G for genetic. So in this situation, let's say that the green pea has the genotype, it's a heterozygous genotype. So it has G and it has the green pod allele and it has the yellow pod allele. Now it's being, the phenotype is green because that dominant allele, that capital G, the green pod color, is masking the effect of the recessive allele, which is that yellow pod color. Now in the yellow pod, the only possible genotype is going to be lowercase gg. Okay, because if there was a capital G, if there was that dominant allele, the recessive allele, which is yellow, would be masked. But because the recessive trait is being displayed, it means it must be homozygous recessive. So now we have the phenotypes and the genotypes. The next step is to write down the gametes that are produced by each. So the gametes that are produced. So this parent, the green pea, will produce two different types of gametes. The first one will be they will be passing on the dominant allele, so G, and we show that these are gametes by encircling the allele. The next allele that this green pea could pass on is going to be that recessive allele, right? Because they also have that recessive allele. In the yellow pea, the only possible allele that could be passed on is going to be the recessive allele. They only have lowercase g, lowercase g. They only have that recessive allele. So the only possible allele that can be passed on in the gamete is that lowercase g. So now we have the gametes. Let's actually go ahead and do the test cross. Now, in the test cross, what we will be finding out is the possible offspring genotypes. So we're going to write here offspring genotypes. Okay, and what we can go ahead and do then is make our test cross table a checkerboard, or it's called also a Punnett square. So we're going to start off with our, going to be like that, and then we're going to have only just one meal. So here we're going to have the gametes of that recessive trait, yeah, okay, and what that's going to be is it's going to be that recessive allele gamete, so it's going to be a lowercase g and it's encircled. Now up here, what we will have is we will have the gametes of this green pea. So we have to write again gametes, and now we have two different gametes. So we're going to have to divide that in two. Then of course we're going to have to actually close this as well. Now excuse the writing, it's quite difficult on the whiteboard. So the green the gametes from that green pea are going to be, as we discussed, the uppercase G, again, encircle it to show it's a gamete, and a lowercase g. Now, when we cross them, so this gamete with this gamete, what is that going to make? It's going to make the genotype GG. It's going to be heterozygous. Lowercase g with lowercase g is going to be GG. So that's going to be homozygous recessive. So then, after we've figured out the genotypes, right, that's going to be a ratio of 50% will be, so 50% will be heterozygous, and 50% will be homozygous recessive. That is the expected result. What we then have to do is we can then write the offspring. We then need to write the offspring phenotypes, okay? The phenotype ratio. So, with the genotypes, what we can see, 50% will be heterozygous and 50% will be homozygous recessive. Now, as we talked about before, homozygous recessive is yellow, and heterozygous or homozygous dominant will display that green trait. So then, what we can write, what we can state is that 50% of offspring, the expected ratio is that 50% will be green, will have that green trait, green pod color, and 50% will be the yellow color. Now, it's important to understand that, yes, this is our expected ratio, but each fertilization is completely independent of the previous one. So when each fertilization takes place, there's a 50% chance that it's going to be heterozygous, and there's a 50% chance it's going to be homozygous recessive. So what that means, if there are two separate fertilizations, and the first time we get heterozygous, that doesn't mean that the next time it's going to be homozygous recessive, because the first fertilization will be a 50% chance of this heterozygous and a 50% chance of the homozygous recessive. The next fertilization again will be 50% chance of heterozygous, 50% chance of homozygous recessive, and so on. Now, the more fertilizations that occur, the closer we get to the expected results. But note that it is not that if we have 50% heterozygous and 50% homozygous recessive, and then there are, let's say, 10 fertilizations, it does not mean that 50%, so 5 of those offspring will be green and 5 will be yellow. No, it could be maybe 7 green, 3 yellow, or it could be 7 yellow and 3 green. It's 50% each time in this case, and each fertilization is independent of the previous one. So make sure you do understand that expected results are not always the same as the observed results. As we increase the sample size, as we increase the number of fertilizations that take place, we will probably reach a closer ratio to that was expected in the genotype and phenotype that we did from the test cross.